So chromosome structure. We first started looking at chromosome structure when we did mitosis. And we're going to review that. And what we're also going to do is add to that, because there is some different terminology that you guys need to be aware of for chromosomes as they pair up in meiosis. And we're not going to look at all the terminology that you need to be able to understand and associate with chromosomes right now, but we're going to introduce some key terms that you'll need to know in order to understand what is happening in some of the phases of meiosis. And so to begin, we're going to look at where we started back during mitosis. And we started with just a regular chromosome. And this is before the S phase of interphase. We'll just say that as of right now, this cell that this chromosome is in G of 0. Okay. Now, it's important to remember that if we take this chromosome, and by the way, this is at the structural level, right? A chromosome is a structure. And we take this and we enlarge it to the molecular level. That's where we're going to see our characteristic double helix, right? That's where we see our DNA, which is a nucleic acid, which is an organic molecule. Also important to remember that if we enlarge that, that's where we would see the 3 components of a nucleotide, because that is the monomer of a nucleic acid, where we would have a phosphate group, we would have a sugar. In this case, it would be deoxyribose. And then we would have one of 4 nitrogenous bases, which would either be A, T, G, or C for DNA. After the S phase of interphase, which is DNA synthesis or DNA replication, different names that mean the same thing, which is typical for life science. After replication, then we get a structure that looks like this. So we now have a replicated chromosome, and there are a couple different names for this as well. This can be called a metaphase chromosome, or sometimes just referred to as an X chromosome. And those replicated chromosomes are held together by a centromere, and they are now called sister chromatids. Remember, when we use that type of terminology, sisters and daughters, we are referring to things that are identical. Also, keep in mind, if we were to diagram this out on a molecular level, when the DNA replicates, it unzips and splits, and each new DNA strand consists of one original strand and one new strand, and therefore we call that the semi-conservative model. So I'm just going to kind of put a divider here, and we're going to say that interphase has already occurred in meiosis. The other thing I'm going to do is I'm going to differentiate with color. I'm going to use red to represent my maternal chromosome. And so we'll go ahead and draw that out. And again, keep in mind that we're saying that replication has already occurred. We're done with the S phase of interphase. And then I'll use blue to represent my paternal chromosome. So again, everything is replicated. We are done with the period of growth and DNA replication. So the other thing, we'll say that this is pair 1. So if we're looking at human DNA, we would have 23 pairs, and we're reducing that down to 1 just to make it simplified and more understandable. So just like with our chromosome structure that we looked at before, we have sister chromatids. And since we have a pair of sister chromatids here and a pair of sister chromatids on the paternal chromosome, we can refer to this whole thing as a tetrad, which consists of 4 sister chromatids. Now, not only is this called a tetrad, but this is also referred to as homologous chromosomes. And here's where I want to spend just a minute explaining what this means. So, homologous comes from the root word homos, which means the same. Now, if you look at these chromosomes, one's red and one's blue, one's maternal and one's paternal. So this individual received one of these number 1 chromosomes from mom and one of these number 1 chromosomes from dad. So the chromosomes themselves are different, but we call them homologous chromosomes, or we call them a homologous pair, or we call them homologs, right back to the principle of different names that mean the same thing. So, what are we saying is the same about these chromosomes? So, in these chromosomes, the DNA codes for the same trait, but maybe not in the same way. And I'm going to explain what that means because it's really important. So let's just get this out of the way. Same and same. So it's the same trait, but they may not code for it in the same way. And this is where the idea behind dominance and recessiveness, and there are other examples, but that's where that comes into play. So let's say for just a moment that we're looking at chromosome pair number 1 on a pea plant. And let's say that that pea plant, chromosome pair 1, was responsible for pea plant height. And so if that was the case, the maternal chromosome may have a section of DNA that we'll label big T for tall. So the maternal chromosome carries a trait or a gene that makes the pea plant tall. We'll say the paternal chromosome has a little t, and that little t is what makes the pea plant dwarf or short. So these are homologous chromosomes because they code for the same trait, but they can code for the trait in different ways. And we're going to learn that those different ways can be called alleles later on, but that clarifies what it means to be homologous chromosomes.